All right. Hello, everyone. I'm really excited today to build on the previous two talks that you just saw and discuss how we're leveraging this common technology platform, which was just introduced by Dr. Esselman, for robotic, automated invasive muscle density estimation. So invasive muscles don't need much introduction, but they're widely considered to be among the most consequential biological invasions North America has ever seen. Since the very first zebra mussel was identified in Lake St. Clair in 1988, they've now proliferated to the point that they represent approximately 90% of all animal biomass at the benthic zone or the floor of the lower four Great Lakes. Just to put that amount of abundance into perspective, it's further estimated that they have the ability to filter all of the water in Lake Michigan every 2 weeks, filtering that water, depleting it of essential zooplanktons and nutrients critical to the food web and fish food chains. So it's difficult to reliably control what we cannot measure. So our scientific knowledge of mussel populations is currently severely constrained by the existing sampling approaches that we use for monitoring them, namely small shipside dredges and scuba dive expeditions. These tools, while highly accurate and provide a gold standard of, we're retrieving a physical sample from the bottom, we know exactly what was there, they sample an incredibly small area of less than a quarter of a meter square. Furthermore, as quagga mussels are known to be sort of shifting their habitat into greater depths, it becomes increasingly difficult to safely scuba dive to those sites. Meanwhile, the small dredges cannot effectively sample. They cannot bite into the rocky, hard substrates which are favored by mussels, which is where we would, most like to measure their population. As a result of these gaps, this results in management blind spots and kind of a void in the scientific literature on their changes in population over space and time and our ability to effectively intervene. So this work is leveraging the common technology platform that was just introduced by Pete and Phil. Exciting thing about this platform is we're getting very, very clear ground images on the bottom, which let us actually visualize and count mussels that even though they're only the size of a fingernail. And so, we have the exact sort of physical footprint of this area, and we can assign it to kind of a world coordinate system based on the AUV's advanced sort of dead reckoning ability and its ability to monitor and maintain a consistent height above the bottom. And so this sort of depicts what an individual sample looks like from this tool. And I've overlaid kind of a typical quadrat sampling frame that would be used in ecological studies. So every second we're collecting roughly an order of magnitude kind of larger aerial sample compared to what you would have, let's say, a scuba dive or the small dredge. And then this is sort of a look at one of our data sets from our upcoming publication that was collected near Muskegon. The AUV is essentially programmed to fly a dense we call it a mowing the lawn pattern. And then you'll notice there's certain so each marker indicates a single photograph that's taken, and there's some missing entries there where the images have not met our image clarity kind of screening metrics. So there's sort of a preliminary step before we hand this off to the automated software is the image clear enough that a human observer could reasonably see what's in the image. So the way we developed this tool is this is sort of what this is a look at what a typical human annotator would see. And they essentially have to painstakingly go in and dot annotate or trace the individual mussels on a computer screen. This process can take anywhere from 15 minutes to 60 minutes, depending on scaling with the number of mussels. It takes longer. But fortunately, once this is complete, we can then synthesize those judgments into a repeatable software and that's what we're doing with machine learning. So essentially, we're taking that, once the model's trained up, that up to 60 minutes of personal effort per image is now reduced to under a second for the trained model. And so the data can be processed concurrently as it's being collected with the AUV, essentially. So the output of this model, which we were very creative, we named it Mussel Finder, is depicted in the pink shaded overlays there. And so then via sort of a regression relationship, we can relate the percentage of the image covered in this sort of pink highlighting, the mussel percentage coverage, to its, this is a very good predictor of abundance and density because we also know the exact area of that image, which depends on just the height of the AUV and the camera parameters. So that was slowed down for visualization purposes, but transitioning from still photography to video kind of better demonstrates the real-time capabilities of this approach, and to sort of scale to like higher endurance, kind of continuous transect mapping. So this sort of takes us, well beyond what is sort of feasible with human effort alone. And then this is another sample. This is in Lake Michigan, and this also really illustrates the patchy nature of mussel distributions, where if you were to drop your dredge off the side of the ship and it falls into one of these sandy areas, you'd get an incredibly low, you'd be deceived into thinking there were no mussels there, when in fact, if you kind of integrate it over a larger area, you would see that there's considerable density. So in our upcoming publication on this sort of a methods paper describing this approach, we've benchmarked its performance in different conditions, representing different sort of environmental limitations. So in this plot here, on the horizontal axis, you have the abundance estimated by the algorithm, and on the vertical axis, you have the true abundance based on human visual estimates. And so we see, not perfect, but pretty strong agreement, even in conditions with considerable vegetation and distracting shells and other nuisance elements on the bottom. If we go to clear images with good water clarity, the accuracy and precision are even higher, above sort of a 90% threshold. And just to sort of put that into perspective, Environment Canada did a small study where they physically sampled and removed mussels from the lake bed and counted them in a lab. They then distributed the mussels onto sort of a well-illuminated flat background, sort of a light table. And in sort of a pristine lab setting, the relationship between the percentage of the image occupied by mussels, sort of their pixel-based count, and their abundance was sort of at a similar level of performance. And so this essentially implies that in favorable conditions we're achieving lab level accuracy in the wild, which we think is pretty nice benchmark. So we've begun operationalizing this research already. One example is the Great Lake Commission's SWIM project for spawning whitefish invasive mussel initiative. So in this example, the AUV again flew that dense lawnmower sort of style pattern over Good Harbor Reef, which is believed to be ecologically significant for whitefish spawning. And requires further work just to really validate the absolute densities being predicted by the model. However, the relative trends that we see seem to sort of agree with kind of expert expectations based on changes in substrate and depth, so on the figure, the darker blue dots indicate higher abundance, and the lighter values indicate lower abundance. So we know, for instance, mussels prefer a harder substrate, and you can see their abundances fall off over that sandy area. So this approach cannot really be considered a static tool in the same way as a dredge that is lowered from the side of a ship. So it's involves kind of a complex orchestration between hardware and software, and so it really benefits from continued development. One of the limitations of the model currently is we're only really looking at that top surface expression layer of the mussels, whereas we know that they tend to form dense clusters and even pile on top of each other and achieve significant growth kind of vertically off the lake bed. So to mitigate that, we're really excited to kind of start integrating the structure for motion type approach that Phil just mentioned, or additional cameras to better capture the height of the mussel bed. Furthermore, in our accuracy metrics are relative to human-based counting of mussels, sort of what can be perceived visually. We don't have hands on the ground to it's not considered kind of gold standard physical sample. However, this error has sort of been benchmarked in the 3 to 20% range by previously by Karatayev and others in Lake Erie. And so and we believe that by pairing the AUV with additional kind of targeted or random physical sampling, these we can better kind of validate that we're predicting the correct absolute abundance and not just relative trends. Lastly, because we're relying on color imaging, this cannot see through dense vegetation. However, fieldwork could be coordinated to sort of mitigate some of that difficulty. Sort of shoulder seasons, where there's not as much vegetation in the water and water clarity is improved. Another limitation is we're currently only predicting a single class, which is the live mussels. However, the model has the ability to differentiate live versus discarded shells, and so that is we're only really limited by what the human annotators put in the training data, essentially. So that can be expanded, and then, like you may have heard with a lot of AI, it benefits. It's continually learning and benefits as the size of the training set increases. And so this really should be considered like the first version of this model. We're excited to develop it further. This is just one example of kind of showing its ability to differentiate those live mussels, which tend to be characterized. They're circled in the yellow circles there. They tend to be characterized by a shell gape feature, that sort of dark visual slit. Whereas we've actually trained it not to detect empty shells, which sort of tend to lie more on their side and are often bleached. That might seem like kind of a trivial detail. However, it's known that because there's so many mussels in the lakes, they're continually dying off and depositing their shells behind. This is actually forming a new, really ecologically significant substrate in the lakes that fish tend to seem to prefer for sort of its roughness for depositing eggs. And so the approach, there's sort of a straightforward extension to outputting this endpoint as well as the live mussels. That's sort of an example, just highlighting the shell hash in that image there. So to recap the method's technical benefits, compared to sort of our previous study in 2022 that was based on scuba diving, the area per sample is improved by over an order of magnitude. The time taken to analyze that data is reduced from 900 seconds, which I believe about 15 minutes, to under a second. So that's a 3 to 4 order of magnitude reduction. And then finally, our accuracy is also increased from around 71% range to a 90% level. So takeaway is we can analyze 4 orders of magnitude more data in the same kind of unit of time effort. So I'll conclude with kind of beyond the technical benefits, we're most excited about the potential application of the method for ecological benefit. So for things like predator management, the food web modeling that I mentioned, kind of targeted inspections around, say, power plant intakes or drinking water intakes, measuring kind of going in before and after control efforts, and then also early detection of previously uninvaded habitats, such as areas in Lake Superior or smaller water bodies. So with that, I'm happy to take any questions.